Michael Snyder, Chair, Department of Genetics, Stanford University School of Medicine, will now take the floor.
Well, it's truly an honor to be here, especially on this particular occasion, opening the university. I think a lot of what I'm going to present will actually build off the themes you heard about earlier. So we believe medicine is broken. It needs to be fixed in many different ways. And for one thing, it's very focused on treating people ill rather than keeping people healthy, as you heard about earlier. Second, even when you practice medicine today, there are many aspects that we think are obsolete. And I think you, as new PhD students and M students, will be in a position to actually transform the way medicine is done. So if you think about it, the way we do healthcare today, even when you practice healthcare, is that you get in a car, typically, drive, maybe here you get in a subway or a tram, and drive a long distance, often at a very inconvenient time. You wind up at a doctor's office that looks pretty much the same as it did 40 years ago, maybe with a few new gizmos. They'll put a needle in you and draw large amounts of blood, and sometimes that hurts and people pass out. Even from that large amount of blood, they'll typically make very few measurements, and then they'll also make some physiological measurements as well. And perhaps the last part that I think needs improvement is that they'll make decisions about your health based on population-based data rather than individual-based data. And to illustrate this last point, I know I'm in Fahrenheit here, but you can see centigrade on the left side of this. We've been taught since we were little that the temperature when you put a thermometer in your mouth is 98.6. But if you actually look at the data out there, it's more like 97.5. I myself am 97.3. But the more important point here is there's a spread. The 25th quartile is 94.6, and the 75th quartile is 99.1. So in today's world, if you travel to a physician's office and they measure your temperature and it comes out at 98.6, if your baseline was 94.6, you're actually, they'll tell you you're healthy, but you're really up 4 degrees over your baseline. And we would argue that you're probably not healthy if you're up 4 degrees over your baseline, something is off. And so a big theme of our work is to try and understand what your individual baseline is for every person, so you can detect disease at its earliest time and see these shifts. And we think that knowing that, along with big data, will transform the way we do healthcare. So your health, I'm sure you know, it's impacted by many things. Your DNA is certainly one part of this, but the environment and your activities, lifestyle, have an important part as well. So exercise, food you eat, stress, all these different exposures, environmental exposures. We can now measure these sometimes quite well with wearables, sometimes still pretty clunky, but together we know they impact your health, and we think, again, you can quantify some aspects of this, but you can quantify the impact on people's health by actually now doing deep data measurements on people. And this is a big part of our theme, and I think this is going to lead heavily into AI-driven medicine in the future. And so as many of you know, there's been a revolution in genome sequencing. This year, it'll probably cost about $100 by the end of the year to get your genome sequenced. Now, the interpretation of that's going to be more than $100, possibly $1,000 or more, but it will come down over time. On top of that, maybe it's a little bit quieter, but you can actually measure thousands of molecules in your blood and urine using developments in mass spectrometry and other methods so that we can get a much clearer picture of your molecular composition. Then also, relatively recently, wearables have come onto the scene as powerful health measurements. Just out of curiosity, how many of you are wearing a smartwatch or a ring? Oh, pretty decent number. Yeah, well, I just gave this talk actually at an exercise meeting at Stanford, and 90% of the people all raised their hands. That's a Silicon Valley phenomenon. But I do think it'll become common everywhere, as you'll see. At least I hope I convince you that'll be the case. Okay, so a number of years ago, we decided to see if we could try bringing big data into healthcare. And this is a research study, but you'll see how we're spinning it off to try and get out to the broader community. So we started this project, we call it Personal Omics Profiling, and it's on a relatively small number of people, 109. I'm one of them to start this off. Well, what we do is we'll sequence their DNA, their genome, and then we'll make as many molecular measurements as we can out of their blood and urine. So we'll measure their transcriptome, their methylome, their proteome, which means all their proteins, transcripts, epigenomes, the methylome. We also follow their metabolome and their lipidome. Most of this is out of blood, so out of peripheral blood monocyte cells for the transcriptome. The other homes are measured out of plasma or out of urine in the case of metabolites and lipids. We'll also measure people's microbiome from their nose and from their gut, which many of you know is pretty important for people's health. And then this is on top of deep clinical testing, various questionnaires, and some advanced tests like stress echocardiograms and various glucose control measurements. You'll see why in a minute. And then we also, when wearables first came out as fitness trackers, we realized they're pretty powerful health monitors, so we put those on people too. So you might say, why are we doing all this? Well, we're trying to understand what it means to be healthy, not what it means to be sick. What does it mean to be healthy? How does that compare amongst different people? How does it change over time? What happens at the earliest times of illness? I forgot to say, because what we're doing, we're collecting big data on people, but we're also doing it longitudinally. So we'll sample people every 3 months while they're healthy. And then if an adverse event like a viral infection comes along, we'll take 5 to 7 additional samplings. So it's not just big data, it's longitudinal big data. And so, as I say, we can actually follow how it changes over time. What happens at the earliest time of illness is like a viral infection. And one of the most important aspects for this group is, can these advanced technologies like genome sequencing and such be powerful for better managing people's health? And the answer with regards to this last part is yes. Just from the first 3.5 years, 49 cases, about 47 individuals, learned something pretty important about their health. So for example, and it spanned a wide range of areas, so heme-oncology, cardiovascular disease, metabolic disease, and so on. And some of these were a pretty big deal, meaning we caught some with early lymphoma, 2 people with pre-cancers. You see MGUS, smoldering myeloma. I don't know if I can point. Well, anyway, you can see them on the slide. We had 2 people with serious heart issues. All these were found pre-symptomatically. So we like to think about this as that if your health is a 1,000-piece jigsaw puzzle, the way we practice medicine today is we take about 6 to 8 of those pieces. We're trying to take about 800 pieces, so we get a much clearer picture of people's health. And as I say, these were all found pre-symptomatically, and they were all found with different technologies. No one technology found all of these. And sometimes it's a combination of both. So, for example, one of the cases there were 2 serious heart conditions. One was picked up by genome sequencing, another through wearables. Imaging found the early lymphoma along with some other biomarkers and so on and so forth. So different technologies reveal different things. So just to go in this in a little more detail, genome sequencing alone found 12, actually 13 cases that were pretty important. So it's not a majority of cases, but if you're one of these cases, it's a pretty big deal. This is what's called the Mendelian disease case. So I'm sure all of you are familiar with BRCA mutations, which puts women at high risk for breast and ovarian cancer. We did have one individual who had this, who actually was an orphan, so they didn't know their family history or who their parents were for this. We had 3 of these individuals had, again, something very related to this on their health. So for example, that case of RBM20, that's a mutation in a cardiomyopathy gene. It turns out this is a fairly young fellow. He winds up having a cardiomyopathy that was revealed after sequencing his genome. In fact, his father died of a heart attack in his 60s. His aunt had one in her 50s. So it turns out he did a stress echo follow-up, and sure enough, he actually does have this heart defect. He's on medicine now, all revealed from his genome sequence. At the top, one of the individuals had a mutation in that gene, SDHB, which puts them at high risk for certain kinds of endocrine cancers. And sure enough, they did a whole body MRI follow-up and had early thyroid cancer. That part was removed, and in fact, they maintain most of their thyroid and don't need replacement therapy, at least at this point. 9 individuals coming in the study were thought to be type 2 diabetic. One of them is actually down near the bottom, that HNF1A. It turns out that's a MODY mutation. So the person's been a MODY mutant rather than a regular type 2 diabetic, and they've been on the suboptimal medication for years. It's actually shifted after we sequenced their genome. So the point is, once again, even though it's a minority of cases, if you're one of these individuals, it's a pretty big deal. Now, this is what's called Mendelian disease, where a single gene mutation puts you at high risk. And so again, this was powerful. Most diseases are actually complex disease, and I won't have time to get into this. I'm not sure how many of you are computer scientists anyway, but with these new machine learning methods, we can actually get at the genetic basis of disease at a level that's never been possible before. So even something like this AAA, which is usually discovered when people's aorta bursts, and they die 90% of the time, we can actually get at the genetic underpinnings by using, again, some of these computer science methods to be able to dissect the genetic role of disease. It was a combination of genome sequencing and machine learning. We actually went from 3 candidate genes to 60 genes that all look quite right. On top of this, for ALS, there were 7 genes known from what's called a genome-wide association study. We found 700 genes. These methods are really, really powerful for uncovering the genetic basis of disease. And in this first case, AA, we can actually build predictive models that are as good as some of the best polygenic risk scores that are out there for predicting who's at risk just from a genome sequence. So we think, again, these methods will be very, very powerful for predicting disease risk. Imaging turned out to be powerful, I mentioned already. We found one individual with lymphoma. It turns out they had a very enlarged spleen. They also had several blood markers elevated that said something was off. So in fact, we dug into more detail, and sure enough, they did have the lymphoma. And that person was, again, this was all caught pre-symptomatically, and that individual has been treated and actually is cancer-free since that happened about 7-8 years ago. At the bottom is an individual, also a pretty young person. Forgot to say overall the cohort is older, 53.4. And so the chances of people being at risk for one of these things is much higher than if, say, you're in your 20s. But nonetheless, it did expose things on some fairly young folks. In this case here, there's one individual who had way too much IgM. His 2 samples were way, way above everyone else's in terms of levels of IgM. And sure enough, follow-up tests said he had this early pre-cancer called MGUS that can convert to aggressive cancer. And so he's getting watched much closer. I think there are other avenues one could take to actually take a more aggressive standpoint on how to better prepare for any disease that might come later. We're very interested in metabolic disease because it turns out I'm type 2 diabetic. And it was in fact predictive from my genome sequence. I'll tell you a little more in a minute. But coming into the study, I mentioned 9 individuals were thought to be diabetic. Well, it turns out 2 people were diabetic who didn't know it when we measured them, and many people are pre-diabetic. That's that orange box in the middle. And it turns out, you may know this, but 90% of pre-diabetics have no idea, and 70% of those will go on and become diabetic. So I guarantee a lot of you are probably pre-diabetic and don't know it unless you're getting measured. During the course of the study, just from the first 4 years, 9 other individuals were classified as diabetic, and others had diabetic range values. So the numbers went up and more became pre-diabetic. And we're very curious, how do people become diabetic? Do they just gradually get there? Does something spike it and trigger it? So it turns out of the 9 individuals who became diabetic during this profiling, 7 of them just gradually got there. And the 2 at the bottom are cases of that. Why don't you look at the middle box? The orange is fasting glucose. The gray is hemoglobin A1C. That individual gradually went up on both, was classified as diabetic and got treated, so their numbers dropped. The bottom is OGTT, same thing, that this individual gradually got there. 2 other people got there by a spike, something as though something triggered it. The top one is me. That's 8 years of data. I first became diabetic, if you look at that first spike on the left, after a nasty viral infection. It was RSV, which is actually flying around right now. And so I was kind of surprised. I went in for an insulin resistance test, and sure enough, just measuring my fasting glucose, it had spiked up. It was 3 weeks after that infection. And I know that looks like a tiny blip. That's about a year of data. In my case, I was able to change my lifestyle. I cut out all sugars, doubled my biking, and started running, and I got it down to normal levels. And in fact, it was so boringly low, I stopped looking at it. I did the perfect control experiment, but you'll see it spiked up again later. Do you see that 7.1? Someone else spotted that. So Mike, do you realize your hemoglobin A1C at the time was 7.0? It was just before that. So no, I had no idea. And we looked back, and you can see it spiked up again. Guess when I stopped running, it was right at that spike time. I also got a second viral infection. That was a rhinovirus. So we don't know if it's the running or the viral infection. I suspect it's a combination of both. But I did start running again. I never brought it all the way down, but I did get it at least partially under control for a while. And then it just gradually kept going up. And I was running about as much as I could. My schedule could handle about 4 days a week, 4 to 5 miles a run, and yet it kept creeping up. And so I decided to try something different. I switched from running to weightlifting. You can't tell, but I've been lifting weights for 6 years now. And actually, that failed. The idea was that muscle mass is better for glucose homeostasis. And that actually did not work, partly because it turns out the way I'm diabetic. In hindsight, should have figured that out. Come to that in a minute. But I kept going up and up and up. And so finally, I bit the bullet, and I took metformin, which is the frontline drug for type 2 diabetes. And guess what? I'm a non-responder. So I went all the way up to 7.5 off the chart here. And so what was the solution? More data. So it turns out that my glucose disposition is very, very slow. I actually make insulin. My cells respond to insulin. I just don't release it from the pancreas. And there's a drug for that. It's called a rapinolide. And that works beautifully on me. So that actually worked. So the bottom line is we call this precision diabetes by actually knowing better what's wrong with you. Diabetes is probably at least 5 different diseases, 5 subtypes, all which are treated somewhat differently. So by understanding better what was wrong with me, we could treat that. And I think this could be done for everyone. And by the way, these days I've switched from rapinolide to these GLP agonists. You may have heard about them. Everybody's using them to lose weight. They really do that. I actually don't want to lose weight, but they do have you lose weight. In fact, the Hollywood scene's all using these. It's hard to get the drug in the US sometimes now for diabetics because it's become so popular as a weight loss drug. Anyway, that's a bit of a side. So the point is, again, with more data, I think we can measure people. A lot of this stuff can be done more routinely. This is still a research project, but I think it can be executed better. The other thing we can do with this is because we're following people over time, we can see how they age. And it turns out that everyone ages differently. I'm the one on the left, so I'm a typical ager. My coagulation, metabolic systems all alter with time, not in a favorable way. The person on the right, it turns out they're a cardioager. Their cardiac hypertrophic signaling pathway is actually shifting over time. We later learned this person's stage 2 hypertensive, which sort of makes sense with that general idea. In the end, we had enough data that we could actually classify people into what we call ageotypes, different patterns of aging. And so each row is a different ageotype. I know there are more than 4, but I'm just showing 4 is all we were statistically empowered for. Each column is a different person. So the 4 people over to the right, they're aging in all 4 categories. They're kidney, liver, metabolic, immune agers. The one in the box on the right is aging in 3 of the 4. Not much of a kidney ager, but is aging in the other category. And on the left, you can see a person who's a liver and metabolic ager, and in the box next to that is a kidney ager. So people are aging, again, differently, sometimes in one, sometimes in multiple categories. And there are clinical markers associated with these, and you can actually see which ones are shifting, and then we think you'll be able to treat people according to the way they're aging, as well as just understanding better, catching things pre-symptomatically. And again, I think this can be done with a lot less data. We used a lot of data for this, but I don't think you need that level of data to see how people are aging and then actually treat people accordingly. One thing I get asked a lot is, how are you going to scale this? Well, I'm in Silicon Valley, so the answer is form a company. Okay? I think academics are really great at discovery and proof of principle. We're not that great at scaling when you get right down to it. This is what companies are good at. So we formed a company called QBio that I should say everything I'm telling you is totally conflicted, so you can distrust me if you want. But just from the first 100+ people that the company started profiling, they found a number of health-related discoveries. Again, all pre-symptomatically. Now, they're doing a medical version of what I presented. So they're not doing RNA-seq and these things that the medical establishment won't necessarily know how to interpret, but they're still collecting a lot of big data in clinical values, and they also do whole body MRI. Now today, I'm sure, probably even here, every physician I've ever talked to at Stanford says you absolutely should not do whole body MRI because people have nodules, right? Women have them in their ovaries, men have them in their pancreas, or sorry, in their prostate. Mike Snyder says you absolutely should do whole body MRI. Has nothing to do with whether you have nodules or not. The question is, do you have growing nodules? And the only way you're going to know that is longitudinally. So I think in the future, as these things get inexpensive, we should be able to do this for everyone. And this is how a lot of these discoveries were made, often by MRI, but also by other markers as well. So we caught some with early stage ovarian cancer, prostate cancer, cardiovascular disease. We even caught some with early stage pancreatic cancer, which is almost never found early. And so again, these deep data dives do give you a picture of people's health that we think is important for better managing their health. Okay. The other aspect that we're trying to change is transform the way healthcare is delivered, at least many aspects. So we're very big on trying to Amazonize healthcare, if you will, if that's a verb. We are trying to bring in wearables and actually remote sampling as a way to better measure people in their native environment. So with regards to wearables, you probably know these are pretty powerful. They're becoming very popular. As I say, we got involved in these a number of years ago when they were just fitness trackers and thought they could be great health monitors. They're powerful because they measure you 24/7. As long as you keep them charged, they'll follow your health basically 365 days a year. And they measure all kinds of valuable things these days. Pretty much all the rings and watches will measure heart rate variability. Some of them measure skin temperature, some blood oxygen, some blood pressure, some galvanic stress response, which is not yet in the healthcare system, but actually could be. It has useful data on it. So they collect a lot of very, very valuable health-related data. And so we actually decided to try these. We put them on our cohort to see what we could learn. And it turns out that one thing you can learn is you can detect infectious disease very early. Now, the way we tend to detect infectious disease early, sorry, the slide's a little out of place, but it's all right, is that we typically use a thermometer, which is a 300-year-old concept. And it turns out that works well for some things, but doesn't really work that well for COVID, right? Half the people don't get a fever. So actually, these days, PCR is a better test, but you don't do that all the time. Antigen testing is obviously reasonably effective, but still it's imperfect and you don't do it continuously. So we think wearables will be fairly powerful. And the way we got into this, I may have lost the slide somewhere. Oh, here it is. I skipped it. Sorry about that. So the way we got into this is when we discovered you could detect Lyme disease, of all things, with a simple smartwatch and pulse ox. I forgot to say I'm a big believer in this. I'm wearing 4 smartwatches right now. This is my exposometer, my hearing aids. They are for hearing, but they also help me track activity and other things as well, my interaction score. So all these things you can use for passive monitoring, which we think is very powerful. So my Lyme disease story, it was me who got the Lyme disease. It was found because I basically was flying to Norway, and on that flight, it turns out your blood oxygen drops on airlines. Most physicians know that, but most flight attendants, most passengers don't. It's the reason you get tired on aircrafts. And so as I was flying to Norway, 2 weeks previously, I'd been in an area which is very heavily Lyme infested in Massachusetts. And then while I was flying there, again, 2 weeks later, my blood oxygen was abnormally low, and it never came to normal when we landed. If you look at the top, my heart rate was elevated as well, and I later learned my skin temperature was up. This is all pre-symptomatically. I did get a fever off and on. I went to a physician in Norway who drew blood and said, yep, you've got a bacterial infection. He recommended I take penicillin. I said, no, I should take doxycycline. Got a little tense there for a few moments. He did give in and gave me the doxycycline, which cleared it up. When I got back, I got tested, and sure enough, I was Lyme positive. I had my antibodies and some antigens left. And I had given blood fortuitously 3 days before I left, and sure enough, that seer was negative. So I seer converted during that time. So the point out of all this is that we could first detect Lyme disease pre-symptomatically with a simple smartwatch and a pulse ox. So we retrospectively looked at data, and it turns out that I had 4 times when I was ill, one of which was asymptomatic, and every single time I had elevated heart rate, resting heart rate, and elevated temperature. And the same was true for 3 other people who had gotten sick, one of whom got sick twice. Every single time, we could see they were ill from their resting heart rate. It had jumped up over background, all pre-symptomatically. So then along came COVID, and it was a little dated, but we're still running about 50,000 cases in the US per day, and I'm sure it's way more than that. It's just that people aren't recording it anymore. And we think that these wearables could be very, very valuable for infectious disease detection. So we very quickly, when the pandemic came out, we partnered with Fitbit, started a study and pulled in 5,300 people, 32 of which who had had COVID and were wearing a Fitbit at the time. And this was our very first case. You'll see it, this is resting heart rate on that top graph. Day 48 is when they were diagnosed with COVID. Day 47, the red one, is when their symptoms first appeared. And it turns out their resting heart rate jumped up 9.5 days prior to symptom onset. And so it's a very, very sensitive detector. The bottom is a real-time detection algorithm I'll tell you about in a minute. And it turns out overall, of the 32 people, 80%, we could detect illness at or before symptoms, and it was a median of 4 days prior to symptom onset. And the reason these are so powerful that we can tell, even if you're just jumping up 2 beats a minute over your normal background, you'll pick that up as a pretty strong signal because, again, resting heart rate's a very, very sensitive measure of illness. So we have gone on to set up a cloud-based system for monitoring millions and millions of people. Ideally, I'd like to monitor everyone on the planet. 3.8 billion people have a smartphone, okay, nearly half the planet. And if you were to pair that with a smartwatch, we think you have a health monitoring system for most of the planet, or at least half the planet. And so a real-time detection system, I won't get into it, but it basically follows your baseline, looks for a jump up, it's anomaly signal detection, looks for a jump up in a circadian fashion over your baseline, and if it's up for a certain duration, a certain level, it will actually trigger a red alert. And it does work. This is one of our first cases again. You can see this individual, that's their symptom onset on day 21. They were diagnosed on day 22, but they were getting red alerts starting day 18, 19, and 20 before symptoms appeared. And same number, it works 80% of the time. It works for Apple, it works for Fitbit. I'm sure it'll work for all of them because they're all pretty similar. It does miss certain cases where we have a hard time getting a stable background on people. And it also works on asymptomatic cases. This is one example from Fitbit, where basically that purple day on the right is when they were diagnosed with COVID, and they were getting red alerts for 2 weeks prior to that. Again, resting heart rate is a very, very sensitive measure of illness. I should point out it's not specific for COVID. It's a general stress detector. Turns out workplace stress is number one for triggering red alerts. And I know that's a big issue for physicians, and physician burnout's a big concern. But anyway, that is the number one. I know in the future we'll be able to distinguish respiratory viral infections from workplace stress. Other things will trigger it too, like intense exercise. If you run a marathon, your heart rate will be up for several days. That triggers it. And so you do have to contextualize these red alerts. Most you can figure out, but other infections, here's mycoplasma, other viral infections will trigger these red alerts as well. I can tell you it did work for me personally. I mostly have green days, so it goes off red alert roughly once every 6 weeks. The third and the fifth, I happen to have a family stress event I had to go travel for, so that set off red alerts. And then you can see on the 10th here, I was getting ready to go to New York City, and I wake up, I'm a little bit congested. Is it allergies? Is it, what's going on here? So I do an antigen test, which is in the top right, and it comes out negative. I look at my smartwatch, and it comes out positive, okay, red alert. So what do? I got on the plane to New York City because I listened to my antigen test. Well, it turns out the next day to go to the meeting, you have to get tested. I did a test again, bright positive. So I spent an entire week in a tiny little hotel room in New York City, all because I listened to my antigen test and not my smartwatch. So we think actually these devices, they can be more sensitive than existing technologies. Now, it's obviously not specific. I didn't know I had COVID or I never would have gotten on that plane. But again, I think with more data, it should be pretty trivial to distinguish certain kinds of events from others, especially respiratory viral infections. So again, we think that this is the kind of technology that can move its way into the mainstream. And I can tell you your smartwatch, I won't show you, it actually can follow your hemoglobin levels, your hematocrit levels, even to some extent your fasting glucose and other measurements. We use machine learning to sort this out. It's not clinical grade measurements, but it's good enough to see these shifts from baseline. For the case of COVID, we can also follow long COVID cases. So they'll have elevated heart rate and other parameters that are going on after the COVID infection. We don't yet have good predictive models. We're working on that for this case as well. And Eric Topol has shown something similar. We think this is just one aspect of monitoring with wearables. You probably have heard about the continuous glucose monitors that are out there. These are very, very powerful devices, and especially powerful for following diabetes and prediabetes. What we did early on was started putting these on normal and pre-diabetic people, and what we discovered is that some people do have good glucose control, like the individual at the top. The one in the middle actually is a moderate spiker, and the one at the bottom is a severe spiker. All these were normal by hemoglobin A1C and other measurements. We think that these monitors expose certain aspects of glucose dysregulation you will miss by other assays. And so we could classify people into what we call glucotypes, either low, moderate, or severe spikers. And it turns out there are people who are normal, again, by normal measures, who are just as bad as spikers as diabetics. And it turns out that different people spike to different foods. This is some work from Aaron Siegel. Some people spike to bananas, some to pasta, some to white bread, some to brown bread. All kinds of different things can spike people, and it's all very personal. And so the top case up there, participant 445, that individual spikes to bananas, doesn't spike to cookies. Probably the only person on the planet that doesn't spike to cookies. The one at the bottom is more typical, spikes to cookies, but doesn't spike to bananas. Okay, so again, this is personal, and your microbiome explains part of this, but only a small fraction. We don't fully understand it. Your food is your number one cause of spiking. Okay, we've actually went on to show that people some people spike to bread and peanut butter, others to protein bars, which do have carbs in them. 80% of people will spike to cornflakes and milk. So if you don't remember anything from my talk, don't eat cornflakes and milk. I think it's bad for you. It's probably worse than smoking. It's just not a very good thing. Okay. The other thing we've been working on a lot is this idea about doing remote sampling, so Amazonizing healthcare, if you will. And I know what this is going to sound like, but this does work. Okay. What's done here, we spent about 6 years working on this, trying to figure out the best way to capture blood spots and keep the analytes stable. And so we ultimately settled what you're seeing here are these Neoterix devices, which sample about 10 microliters of blood. We also use TASO devices, which sample a little bit more. And what you do is you collect these samples at home, mail them by FedEx to our lab, and we'll do these deep omics profiles on them. We measure about 2,200 analytes. So lipids, metabolites, proteins, and we have very targeted assays for health-related molecules as well. So again, we can follow this from samples collected in the home. And so maybe I'll skip over this quickly, but we first showed that most of the analytes are very stable. So nearly all of your proteins are stable to storage and temperature. There's a few that aren't. And likewise, for metabolites, most of those are stable as well. Lipids are half are, half aren't, but we can correct for those that are unstable. So the next thing to do was do fun experiments with this. So we had 32 people drink this Ensure shake. I don't know if they sell that over here, but it's all over the US. It's supposed to be healthy for you. It doesn't taste very good. But we sampled people at day 0 and then 4 times after that. Okay, what we discovered, this is not a surprise, hundreds of molecules change, including some pretty important glucose control molecules. If you look at cluster 1, that's the insulin C peptide. You can see insulin on there. There's an incretin called GIP. For those of you who follow glucose control, these are pretty important molecules to measure. They can actually tell you what subtype of diabetes you have, we think. On the right shows some other inflammatory and other markers. The most interesting result to come from this, so everybody's drinking the exact same shake, okay, 32 people. Well, it turns out they all have very different responses to the shake. The top part just shows carbohydrates. The bottom part classifies people. Let's take a look at the top part. Every box is a different person. And if you look at this one here, S13, yeah, I guess the pointer, oops, doesn't work. It's the one, well, right over your head there. That person plummets, their carbohydrates plummet rapidly after drinking the shake. But look over 3 over in the light blue, 821, S21, that person's carbohydrates skyrocket. These are 3 different carbohydrates, and so on and so forth. You'll see people who go way up for carbs, others that go down. So everybody's reacting very differently. The same is true for free acid, amino acids as well. If you look at the bottom part, see the gray dots in the middle? That's the average. We have some people, like we classified them into 5 groups. In group 5, that brown spot there is actually inflammatory markers. The group 5 individuals have an anti-inflammatory response when they drink the shake. Group 4 and group 1, the brown spot is higher than average. They have a pro-inflammatory response. So they're getting inflammation from the same shake. So everybody's reacting very differently, and we can measure that. And we think this is a big deal because, at least in the US, 10% of people have inflammatory bowel syndrome, thought to be induced by food, at least in part. And we can measure this and tell people what foods are doing what to them, again, at a very detailed level. And so I think we can not only follow people at fasting times, which I think is important, we can follow them after various interventions like the food they eat. The last thing we've done here is we've been combining, we just published all this recently, this multi-omics sampling we're getting from microsampling with wearable data. So this happens to be me. We collected 98 samples over just over 7 days. So very, very detailed profiles, roughly every hour while awake. And then, again, combine it with the wearable data. And we look for these time series shifts, if you will. So certain things, causative events obviously occur before, typically, downstream events. And so we can try and follow these, do this time lag correlation, it's called, and look for which molecules are correlated with what, or what physiology. And what we discovered is that there's thousands and thousands of correlations between biochemical markers, as well as between the biochemistry and some of your physiology. Up here is heart rate. That's glucose at CGM and steps. We can see all the correlations, and we can quantify this. And it turns out there's all kinds of interesting research data, as well as useful, we think, treatment data in there as well. So that top panel is alpha-synuclein, which is involved in Parkinson's. And so we can actually see when it's spiking and correlate it with what kinds of events. It turns out it actually spikes before your glucose spikes. That's known already, but never quantified. It's about a 15-minute delay. We can also, you can see insulin and the C-peptide insulin correlates with glucose. The third one down is actually a very interesting immune ligand that goes up about 15 minutes after your glucose goes up. We think this may be important for the inflammatory response you get when your glucose spikes. So again, we can see all these correlations and quantify them. And I think this is going to be very relevant also for quantifying diabetes. So how are we going to deal with all this data? Well, it's like your car. Your car has tons of sensors on it. A race car has 400 sensors on it. And you have a limited number of panels on your dashboard, so it integrates all the information that you're collecting from these sensors. And so we build a dashboard that pulls in your wearable data, your clinical data, your microbiome, and all the other omics data. And you can actually display this at your personal level, meaning you can display it at daily, weekly, monthly, whatever you like, so you can better personally manage your health. I don't think it's going to replace the physician. I think it's only going to help the physician to have this kind of data available, again, all displayed in a useful fashion. So this is Mike Snyder's view of the future of healthcare. You don't have to agree with it, that's fine. But I'm a believer that people will be getting their genomes sequenced before they're born. The technology is there to do that, especially with the cost of sequencing dropping. So you'll be able to see people's disease risk before they're born. And then together with biochemical and wearable data, you'll be able to build up personal AI-driven programs to follow people's health and see how they're shifting over time and minimally catch disease early, and hopefully better manage and treat disease based on data-driven approaches. So I'm very fortunate. Get my lab next. Yeah. Asked to say something about spinning this off. We're a basic science lab. The goal of this is, again, to do research, to discover essentially new principles, show proof of principles. But we do, when we see something interesting, we spin off companies. And so I've founded 17 companies over the years, which is very doable if you have the right infrastructure. It's something I think could be done here, given the talent that sits in this room. Anyway, these are just some of the ones. The ones in boxes directly spun off from the work I just showed you. We weren't intending to form these companies, but once we saw something useful, we thought the way to get it out to the world was actually to do what I just said, form a company and spin it off. And so this is my talented team here. This is the wearables up top when they were at their peak. Microsampling is a group at the bottom. And then on top of that, we have a lot of other people work on the genome analysis. And so I want to thank you once again for having me here. And I hope this gives you what I hope will be a little flavor of the future of medicine. You can see what you think.
È conclusa la cerimonia di inaugurazione dell'anno accademico di Humanitas University. Invitiamo i gentili ospiti ad accomodarsi nel foyer per un light lunch. The opening ceremony of the academic year of Humanitas University is now concluded. Our guests are kindly invited to proceed to the foyer for a light lunch.